Okay, welcome to this next podcast for cell biology. In the previous podcast, we talked about connective tissues and how they secrete this extracellular material in animals and plants, as well as other cells. In this podcast, we're going to focus on a different kind of tissue, epithelial tissue, and how those cells within the epithelial tissues communicate with each other. So let's go ahead and get started. Epithelial cells and cellular junctions. Epithelial cells are a very prominent cell in our body. Of the 200 plus cell types in the vertebrates, most are epithelial. Epithelial cells cover our body as they are the epidermis of our skin, the outer covering of our skin. They also line our internal cavities. So our blood vessels, our respiratory system, our digestive system, any of our cavities are lined with epithelial cells. So these epithelial cells, they must bind with each other in a way similar to this. They can take on many different shapes and sizes, but for now I'm just going to draw them as this single layer of cells. So cells within the epithelium must, as I just said, bind to each other. They facilitate communication. So in the way they bind, they communicate with each other and between one side and the other side of the tissue, and they will allow for the passage of some molecules across the epithelium, but not all. So the way they're held together and the way they communicate is all going to be dependent upon cell junctions. So all of this is linked to cell junctions. So I'm going to draw the cells of the small intestines here, just to demonstrate and explain some of these junctions. So we have one cell here, and it's going to be attached to the basal lamina, a structure outside of the cell. And it's also going to be connected with a neighboring small intestine cell. We've drawn something similar before when we were talking about the transport of glucose from the lumen of the gut to the other side of the cell into blood vessels. So this is kind of like an old friend of yours just coming back from the past. The first one I want to talk about are tight junctions. And tight junctions, I'm going to draw here. Within the epithelium, what they will do is they will prevent the movement of fluid and molecules in between the cells. So they will seal cells together, blocking lateral transfer of fluids and other molecules between the two cells. These tight junctions that span from one cell to the other are made up of two proteins. One's called claudin, and the other is called occludin. And that's all I want you to know about tight junctions. The next several I'm going to talk about together. They include the adherens, the desmosomes, and the hemidesmosomes. All three of them are important in providing mechanical strength. So I'll write that one down here. All three in providing mechanical strength. They hold the cells together in a way that's different than the tight junctions. The tight junctions' whole purpose is just to prevent movement between the two cells. These other junctions hold the cells tightly together. The hemidesmosomes and the desmosomes interact with the cytoskeleton. and more specifically with intermediate filaments. And in doing so, that helps provide this mechanical strength. We call these desmosomes because they interact with desmosome proteins on each cell. These are hemidesmosomes because they involve an interaction between one cell and the basal lamina and not another cell. The adherins bind to actin filaments. All right, that's all I want you to know about adherins, desmosomes, and hemidesmosomes. So let's talk about gap junctions. Gap junctions allow the transport of small molecules from one cell to the next. Molecules like calcium can move through here. It's a fairly small diameter, so you're not going to get large proteins going through, but very small molecules can go through. These are important in establishing that rhythm of muscle contraction for your heartbeat, the rhythm of contraction necessary in the uterus during childbirth, some coordinated behavior that's necessary during embryogenesis. It's important in propagating a nerve signal. They're made up of 6 proteins, and these proteins are called connexins, one of my 2 favorite proteins. Together, these 6 connexins form this channel that allows the movement of small molecules. I would love to teach a whole semester just on gap junctions, but they won't allow me to do that. So that's all I'm going to say about them for now. Well, I'd like to close this podcast with a description of a cellular junction found only in plants, and they're called Plasmodesmata. They're similar to gap junctions, but there are some distinct differences that I think are worth pointing out. So here we have 2 adjacent cells, plant cells. And this opening between them that connects these 2 cells is what we call the plasmodesmata. Just like the gap junction, it allows material to move in both directions. But a couple differences. The plasmodesmata, the channel here is much larger. That's probably the key difference. Larger materials can move through here. It being much larger, for instance, we can get larger material going through, such as pieces of the smooth ER can move through here. Something else that's a little different is that this material outside of the plant cell walls can also move through the plasmodesmata from outside the cell. Gap junctions are held together by these proteins, but this is much more of a open gate here, that anything that is bathing outside of the cell can move in here. This allows for movement between this cell and distant cells. It doesn't necessarily have to be right next to it. In this way, they don't even have to directly cross the plasma membrane. They just move through these openings. All right, so the key difference here, again, just to reinforce this, is that the plasmodesmata openings are much larger and they're continuous with this extracellular space, so that molecules can cross between these cells and adjacent cells without ever crossing directly the plasma membrane. I should also mention larger protein complexes can move through here, and even structures like viruses can move through the plasmodesmata. So they're much, much larger than the gap. Okay, that ends this podcast on cellular junctions. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.